Welcome to Section 5 of Molecular Biology. In this section, we'll be discussing DNA mutations. Let's get started. In the last video, we went through this figure in detail, and we discussed normal translation. In this video, we're going to focus on mutations of the genetic code and see how these can disrupt protein synthesis. This is a figure of the genetic code, which can be found in Section 5 of Molecular Biology. You definitely don't need to memorize this, but you should know how to interpret it. For example, if you were given a 3-base sequence of mRNA, also known as a codon, you'd be expected to know what amino acid would correspond with the codon. If you look at the figure, you can see that the codon CCA would correspond to proline. The only parts of this figure that you should memorize are the stop codons as well as the start codon. So the stop codons include UAA, UAG, and UGA, and the start codon is AUG. We discussed these in the last video along with the mnemonics, so we're not going to spend more time on this now. This is a table which can be found in Section 5 of Molecular Biology, and it provides a nice overview of the different types of DNA mutations. Let's go through each of the mutations now. Silent mutations are caused by nucleotide substitutions that do not result in a different amino acid. For example, the codon ACC codes for threonine, which you can see right here. In a silent mutation, the last base in the codon is mutated from C to G. Notice, however, that this codon still corresponds with threonine. If we look at the genetic code, we can see that both ACC and ACG correspond with threonine. Missense mutations are caused by nucleotide substitutions that result in a different amino acid. In this case, the codon ACC is now changed to AAC, and you can see that threonine now becomes asparagine. Obviously, this is much more problematic because it will ultimately affect the structure of the protein being synthesized. A great example of a disease caused by a missense mutation is sickle cell disease. Nonsense mutations are caused by nucleotide substitutions that result in a premature stop codon. For example, UAC normally corresponds with tyrosine. However, if the C is changed to a G, which you can see right here, then this results in a stop codon. This can be very problematic because it can result in a truncated protein. For example, some muscular dystrophy disorders, such as Duchenne muscular dystrophy, can be caused by a nonsense mutation. These are more commonly caused by a frame shift mutation, but can be caused by nonsense mutations too. Ultimately, this results in an altered and truncated dystrophin protein. Dystrophin normally connects the cytoskeleton to components of the extracellular matrix, and a genetic defect can cause muscle tissue disease. I think silent mutations and frame shift mutations are pretty intuitive, but it can sometimes be difficult to keep nonsense and missense mutations straight. Some students use the mnemonic "stop the nonsense" to help them remember that a nonsense mutation results in a premature stop codon. So stop the nonsense. Okay, moving on. A frame shift mutation is caused by insertions or deletions that cause a shift in the reading frame. This means that many of the amino acids are coded for incorrectly. Let's pull up a figure so you can see what I mean. This figure illustrates a frame shift mutation, which can be found in Section 5 of Molecular Biology. The top part of the figure is normal, and the bottom part represents a frame shift mutation. Normally, 3 bases in the mRNA correspond to 1 amino acid. For example, you can see that ACC right here in the mRNA corresponds with threonine. You can see that the cell transcribes and translates the genetic code with cell machinery that reads the information based upon this idea of 3 bases corresponding to 1 amino acid. If the reading frame gets moved or shifted, then the entire sequence is messed up. For example, in the bottom part of the figure, you can see that the DNA base thiamine right here has been bumped over to the right side of the figure, and adenine, which is shown here with a circle around it, has taken its place. You can also see that the strand of DNA shown at the bottom of the figure is now 1 base longer than the strand of DNA shown at the top of the figure. So as you can see, there's now a base right here, making the bottom strand 1 base longer than the top strand. Notice that the mRNA is now shifted 1 base to the right as well. This completely messes up the corresponding amino acids. Notice that isoleucine right here now becomes tyrosine, and serine is now replaced with a stop codon Frameshift mutations are commonly associated with Duchenne muscular dystrophy and Tay-Sachs disease. Okay, now let's discuss DNA slippage. This is caused by a segment of DNA that slips away from the complementary strand and results in a frameshift mutation. Let's pull up a figure so you can see what I'm talking about. This figure illustrates DNA slippage, which can be found in section 5 of molecular biology. The top part of the figure represents normal transcription and translation, while the bottom part represents how transcription and translation are altered in DNA slippage. Most of the DNA is represented by lines, but I've included several segments where you can see the bases to help illustrate a point in a second. For example, you can see ATT, AGC, and TGA right here. Notice that these are complementary to the template strand, so ATT pairs with TAA. Also notice that the template strand of DNA is transcribed into mRNA and then translated into the corresponding amino acids. In DNA slippage, the hydrogen bonds formed by the pairing of complementary regions is disrupted. This means that a part of the DNA slips away from its complementary region. This is illustrated in the figure right here. In this example, notice that the template strand now has a region that is mispaired with the coding strand. For example, AGC right here is not complementary to ACT. Notice that ACT right here was shifted. Normally, mismatch repair mechanisms would fix this, but these are inadequate in DNA slippage. In this case, the slipped region of DNA is not transcribed, and instead, the mismatched ACT sequence is transcribed prematurely. Notice that at the top of the figure, TCG right here is transcribed into AGC, but in the bottom figure, this is replaced because ACT right here is transcribed into UGA. Ultimately, this results in a large segment of DNA that is not transcribed, which you can see right here, as well as a premature stop codon. In this way, DNA slippage can alter the reading frame and cause frameshift mutations. Okay, the final topic for this video will be splice site mutations. These are caused by mutations in the regions associated with the splicing of pre-mRNA. This results in retention of introns that would otherwise be removed. This is an overview figure of transcription, which we discussed in section 3 of molecular biology. Recall that once pre-mRNA is formed, it undergoes several processing steps, including the removal of introns. You can see the introns right here. Normally, the spliceosome, which you can see right here, removes the introns, but if the intron contains mutations, then the splicing process can be disrupted. Ultimately, these retained introns disrupt translation and impair protein function. Okay, let's do a question. A 5-year-old boy is being evaluated for Duchenne muscular dystrophy. Laboratory analysis reveals a mutated mRNA molecule. The molecule is reverse transcribed into cDNA, which is found to be 976 bases shorter than a control group. What genetic defect is most likely responsible for this patient's condition? From the question stem, we can see that this patient has a segment of cDNA that is 976 bases shorter than normal. In other words, the boy has a 976 base pair deletion. If we take this number and divide it by 3, we can see that it's not divisible by 3. For step 1, you need to know that frameshift mutations are not divisible by 3. Let's draw this out. This will represent a normal segment of cDNA. If we take the number of bases here and divide it by 3, we get 9 divided by 3, which equals 3. Now let's assume we remove one of these bases. Okay, so notice that we've removed a T from the sequence, and now we have the sequence AAT, CGA, TG. If we again take the number of bases and divide it by 3, we get 8 divided by 3, which equals 2.6. Now let's assume we remove 2 of the bases. If we again take the number of bases and divide it by 3, we get 7 divided by 3, or 2.3. Finally, let's remove 3 of the bases. If we take 6 and divide it by 3, we get 2. You get the idea. Notice that both examples A and B result in frameshift mutations, and neither of these are divisible by 3. On the other hand, example C is divisible by 3 and does not result in a frameshift mutation. So again, for step 1, you need to know that frameshift mutations are not divisible by 3. If we return to the question, we can deduce that Because 976 is not divisible by 3, this patient must have a frameshift mutation. Silent mutations and missense mutations would not result in a shorter cDNA molecule. A nonsense mutation would introduce a stop codon prematurely, which could result in a shorter cDNA molecule, but this would be a number divisible by 3. So the answer to the question is D, frameshift mutation. Okay, let's do another question. A segment of the beta-globin gene is shown below. The capital letters represent exons and the lowercase letters represent introns. A mutation in what region shown below would most likely result in beta thalassemia? Okay, from the DNA sequence, we can see that the lowercase letters represent introns. From the choices, only B represents a mutated intron. For step 1, it's very high yield to know that mutations in the introns are a common cause of beta thalassemia. Beta thalassemia is a genetic disorder that results in abnormal hemoglobin and the development of anemia. So the answer to the question is B. You should also know that beta thalassemia can be caused by mutations to the promoter region, but this is not a possible answer choice, so the answer must be B.